students, this is Dr. Prats and welcome to my channel. So since last week we started a series on caries. Today we are going to continue discussing about the etiopathogenesis of caries and see what is called as multifactorial concept when it comes to the etiology of caries. Let's roll. So you already know we follow a questioning format in our presentations. What is the pathology? Why do we need to understand this pathology? These two things we have already discussed in the first part of the series on caries. So today we are going to continue with the how or the etiology. So basically today we are going to understand the role of different parameters which are going to act together to form caries. Of course, followed by a summary of whatever we learned today and multiple choice question, the tips and tricks to answer it. Every lesson has to have an objective, and today we are going to discuss the current concepts of caries, and we're going to discuss the role of microflora, diet, teeth and saliva in the causation of caries. According to the current concepts, dental caries is a multifactorial disease. The factors which are taking part are the microflora, which is nothing but the bacteria, diet, we're going to discuss in detail. Of course, it is a carbohydrate-rich diet, which we're talking about here. Host, which comprises of the teeth and saliva, and the time of contact of the microflora on the surface of the tooth. So all these factors, when they act together, they cause the caries. So let's see the role of microflora and what are the evidences which indicate the role of a bacterial flora. Germ-free animals do not develop caries. So the experiments done on the notobiotic rats did not really develop any caries. Although they were fed with carbohydrate-rich diet, these notobiotic rats did not develop any caries. When antibiotics were fed to animals, they were effective in reducing the rate of caries incidence. So this is another proof that it is the bacteria which is causing the pathology. When the teeth were still within the bone, when they were not erupted or exposed to the external environment, they too did not show any incidence of caries. It was only after they were erupted and they were exposed to a lot of factors like bacteria, stagnation of food, and the viscosity of the saliva around. That's what caused the caries, not when they were protected. And histopathologically, there were microorganisms demonstrated within the carious enamel and dentin in the sections. This is a solid proof that caries is indeed caused by bacteria. What sealed the phenomena was by proving the Koch's postulate. So whenever something has to be discerned as an infection, it has to be transmitted to another individual or another living being. So when an experiment was done, and the white ones are the non-infected mice and the black ones are the infected mice. So when an experiment was done, first of all, when antibiotic was given to the infected female, she was completely cured and did not show any caries. When the plaque and the feces of an infected mice was smeared to the non-infected mice, it gave rise to infected mice. So this is a solid proof that caries is a bacterial infection. So there are some characteristics essential for the cariogenic bacteria. Number one is it has to be acidogenic, meaning it should be able to produce acids, because acid is essential for the demineralization of the tooth. The second is acidiuric. Acidic meaning the bacteria must be able to survive a very acidic condition, because the demineralization of the tooth occurs when the pH falls below 5.5. This we've already studied in the last lecture. It has to have a kind of an adhesive insoluble polysaccharide which is called as glucan. This helps in attachment of this bacteria to the smooth surfaces of the tooth. So it should be able to produce these kind of polysaccharides, these organisms. And not only that, they must also store an intracellular glycogen-like substance called as dextran, because whenever there is a low sucrose diet, the organism must be still able to produce the acid by feeding upon dextran, which is nothing but a reserve polysaccharide. So what are the organisms which are responsible in the caries? The first and foremost organism which is involved is a cocci, and the strain of the streptococci is called as the streptococci mutans, and this is responsible for the initiation of the caries. That is, the caries starts by this organism. Whereas the organism which is responsible for the progression of caries is called as Lactobacillus acidophilus, and this helps in progression into dentin and further beyond. And the last organism which we are going to discuss is Actinomyces, which is responsible mainly for the root caries. So these are the three organisms which are involved in the bacterial pathology of dental caries. Dental plaque, as I've explained to you in the previous lecture of this series, is very important in the occurrence of caries, because this is a biofilm which basically gives an attachment of the organism to the surface of the tooth for a longer duration. If the bacteria is just going to touch and go, it will not be able to cause any caries. It has to stick to the surface of the tooth so that it can release acid in that area and cause demineralization, isn't it? So this dental plaque becomes visible whenever the tooth brushing is stopped for about 12 to 24 hours. You will study this in detail in periodontics. I'm not going to discuss too much. Biofilm basically means that it is a thin film which has living beings within it. That is nothing but the bacteria which we're talking about. This cannot be removed just by rinsing your mouth. You have to brush your teeth in order to remove this. And suppose you do not brush and the plaque is allowed to stay for longer duration, then it undergoes mineralization to form what is called as dental calculus. And this dental calculus can only be removed on oral prophylaxis or by scaling. And this is what gives a prolonged adhesive effect for the bacteria. So this calculus is what helps to retain the acids which are produced by the bacteria at a particular site for a longer duration of time. And this is what allows for the demineralization of the enamel and initiation of the caries. So plaque and calculus have a very important etiological role in the caries. So that was about the microbial flora. Now we will move on to the host factors. The host factors can be either intrinsic or extrinsic to the tooth. Factors which are extrinsic to the tooth we will deal it later. Factors intrinsic to the tooth are tooth morphology, tooth position, and enamel structure and composition. The ones which are extrinsic to the teeth are diet, under which I will be covering the carbohydrate in detail. Saliva, the use of fluoride, and of course the immunity of the individual. So let's see one by one, starting with the factors intrinsic to the tooth. So tooth morphology. So you have studied the dental anatomy and you know the morphology and anatomy of the tooth. So when you're looking at this picture and when you're looking at the pits and fissures on the occlusal surface, it is very easy for you to understand that this must be the most common area for the occurrence of caries. Why? Why is the question? Because it is an easy area for the food stagnation. The bacteria can reside there. It is difficult place for the brush to reach and to clean up the area. So both these factors together will contribute to the occurrence of increased caries in this region, that is pits and fissures. And another area which is more common is the proximal contact, because below the proximal contact the brushes do not reach, and if you are not flossing in that area, then there is food stagnation and it is easy for the bacteria to live there, lurk there, and eventually cause caries. The next factor is the tooth position. If the teeth are in normal occlusion and if they're perfectly aligned, there is less chance of occurrence of caries, whereas when the teeth are malaligned and if it is crowded, then it increases the occurrence or incidence of caries. The third factor is the enamel structure itself. Whenever there is any kind of either developmental disturbance or enamel hypoplasia or hypocalcification, this will affect the rate of progression of the caries. It not necessarily causes the initiation of the caries, but the progression is much faster if the enamel is already affected by some kind of developmental disorder. The next factor is the enamel composition itself. This we will study again later on when we are studying the factors extrinsic to the tooth, where fluoride is discussed. So you will understand there that higher the fluoride, decreased susceptibility to caries. So we will understand that later on why. So let's talk about a tooth which is just erupted, whereas the teeth which has been in the mouth, which is present in the mouth for a long duration of time. So when the tooth is just erupted, it is still very vulnerable for the caries attack compared to an older tooth which has become stronger over the years by the absorption of lot of minerals from the surrounding saliva and even the diet which you intake. So the teeth which are newly erupted are more susceptible to caries is the message from this. Let's go to the factors which are extrinsic to the tooth, and we'll start with the diet, which is very important. What you have to understand from this diet, and here I just want you to recollect the Stephan's curve which I discussed in the first part of this caries series, where we understood that it is not the amount of the sugar which is important, it is the frequency of the intake of the sugary foods and drinks. Because so the more frequent intake of the sugar is going to keep the pH of the saliva acidic, and this is going to increase the incidence of the caries. So if you're going to eat a bar of chocolates, eat it in one go. Don't think that if you're going to eat one piece at a time throughout the day, you're going to get less caries and it is going to do less damage. Eat the whole bar, be happy, be satisfied, and then do not eat any sugar for the rest of the day. And of course, brush your teeth immediately after you eat any such sweets. The next factor which could actually help is chewing a sugar-free gum. And they say that even a small portion of cheese after meals will help protect against the dental caries. Basically, the mechanism behind this is the production of saliva. So the more saliva which is produced after a meal is going to flush out everything which is going to be stagnant in the mouth. You're going to swallow a lot, so nothing remains in the oral cavity. That is the idea behind this. The next is the food which is rich in the carbohydrate content is more cariogenic, there is no doubt in it, compared to the food which is rich in fat or cellulose or protein, of course. High calcium and phosphate in your diet will also reduce caries incidence, because the teeth is going to absorb all this from the diet as well as from the surrounding saliva, and that's what increases its resistance towards the caries microorganism. And some factors which you must know when it comes to the diet is that as long as you're using a raw, unrefined food, which are very primitive, they're going to induce less caries compared to the modern soft and refined foods, which are basically sticky because they all go and stick to surfaces. And because they're refined, it is very easy for the bacteria to break it down and cause caries. So go to raw, unrefined foods and go more organic. Let's move on to the factors which affects the karyogenicity of the carbohydrate itself. The type of carbohydrate, I've already told you, the refined one is bad, and especially the sucrose, because sucrose is the type of carbohydrate which is actually helping the bacteria to produce dextran and glucan. You already understood what dextran and glucan are. So glucan is what is responsible for attachment of the bacteria to the tooth surface, and dextran is the intracellular polysaccharide which the bacteria is going to rely on whenever there is a low sucrose diet. So it is the sucrose which helps the bacteria to produce both the dextran and glucan and eventually cause caries. So avoid sucrose, go for complicated sugars. The amount and frequency I've already told you. Frequent feeds of small quantity of sugar is bad. Big amount at one go is better. The consistency, it is again very logical for you to understand that it's the liquids which are better compared to the solids, because liquids are cleared faster from the oral cavity, whereas solids, which are especially sticky, is going to stay longer in your oral cavity, and that is more karyogenic. Genic is basically producing, so when I say karyogenic, it is producing caries. Of course, I said that the liquid form of sugar is better than the solid, but be very, very careful, because you will never know how much of sugar you're taking in. And these drinks, the sugar aerated drinks, have a lot of other side effects other than caries, which you must be aware of. Just don't go blindly and start taking a lot of fluid sugar intake instead of the solid ones. You must know what you're doing. So what is the evidence for this? Caries was not induced when sugar was given to an individual either through IV or by a stomach tube. It was induced only when the sugar was taken intraorally. So this is a solid evidence that the intake of sugar through oral cavity, which is stagnated in certain areas of oral cavity, which is later putrefied and fed upon by the bacteria, which releases acid, is what going to cause the caries. So this we have already discussed several times, that sucrose in sticky form is more karyogenic. Let's move on to the factors which are extrinsic to the tooth, starting with saliva. You know, saliva has a very important role in our oral cavity. It has many functions, but the most important function when it comes to caries is the buffering power of saliva. You know that saliva has important buffering systems, which basically maintains the pH of the saliva in an alkaline area, that is around the 7. The rate of flow, composition, as well as property of saliva also has an important role. If the salivary flow rate is good and if the consistency of the saliva is watery, then there is low incidence of caries. But if the salivary flow rate is reduced and if the saliva is viscous, then that causes an increased incidence in caries. So the buffers which I talked about were the bicarbonate buffers, which is an important buffer of the saliva. And like I said, a rapid flow rate with greater salivary buffering power is associated with low caries activity, is the gist of it. So in cases of xerostomia, which is nothing but a reduction in the flow rate as well as a thick viscous saliva, in cases of salivary gland aplasia, in cases of several treatments of head and neck tumors, Sjogren syndrome. There are several scenarios where xerostomia could occur. So because of this, there is an increased incidence of caries because of the thick viscous saliva which is stagnant. And whenever there is a rapid flow rate, like in case of Down syndrome, there is a rapid flow rate of saliva, and that results in a low incidence of caries, which is again very interesting. Why the rapid flow rate and low viscosity helps in the decreased incidence of caries is because it basically assists in clearing the food stuff from the mouth quickly, and the opposite is true when the saliva is more viscous, because that's going to make sure that the food stays within the oral cavity and is stagnated. Calcium, phosphate and fluoride, which are present in the saliva, help in remineralization process of the tooth to a certain extent. So there is a post-eruptive exchange of calcium and phosphate ions throughout the life between the enamel surface and the saliva. And this is the reason why I said the newly erupted teeth are susceptible to caries than the teeth which are present in the oral cavity for a longer duration of time. Also, saliva carries fluoride, which can have a very carious resistant effect on the tooth. So we'll discuss more about fluoride. Fluoride can be either supplied by community water supply in countries where people drink tap water, and by drinks like tea where fluoride is present, and seafood like salmon is very rich in fluoride. If none of these are available, say for example, people are living in a country where community water supply is not there, they're drinking bottled water. If they don't drink tea much, or if they're not consumers of seafood, then the dentist can apply a topical layer of fluoride. This is going to turn the teeth resistant to caries for a while, especially in the areas of pit and fissures. So the fluoride which enters the body is going to form what is called as Fluorapatite crystals. So these Fluorapatite crystals are less soluble than the hydroxyapatite crystals whenever there is an acid attack, and the concentration of it increases on the outer surface of the tooth, which protects it from the caries and makes it resistant to caries. So like I always say, do not get overwhelmed, take a deep breath. What we studied today are the 4 factors, which is the current concept of caries, that it is a multifactorial disease where bacterial flora, host which involves the tooth and saliva, diet which is rich in carbohydrate, and the time of contact of the bacteria with the surface of the teeth. All of this together will cause caries. Under the bacterial flora, we saw that it was Streptococcus mutans, which is the initiation agent, Lactobacillus acidophilus progression agent, and actinomyces, which causes the root caries. Under the host, we discussed the tooth morphology, tooth structure, enamel composition and structure, and those are the factors which are intrinsic to the tooth, and factors extrinsic to the tooth were saliva, fluoride, immunity, and diet. Under the diet, we mainly discussed about the carbohydrate-rich diet, and under carbohydrate, the sticky form versus the non-sticky form, refined versus unrefined, and solid versus liquid, and the frequency versus the amount. So these were the factors which were discussed under the diet. And the time of contact is influenced by the production of dextran and glucan, which are helpful in attaching the bacteria to the surface, the smooth surface of the tooth rather, and provide a form of attachment so that there is a longer effect of the acid which is released by the bacteria on the surface of the tooth, which will result in demineralization. And dextran helps the bacteria to survive whenever there is a low sucrose diet. So that was the summary of whatever we studied. So an MCQ which can be asked from today's lecture is actually a multiple response question, not a multiple choice question. So the statements that are true regarding dental plaque, there is that it is a biofilm that becomes visible when toothbrushing is stopped for 12 to 24 hours. It cannot be removed by rinsing. It can only be removed by toothbrushing. It undergoes mineralization to form dental calculus. It helps to retain acid in a high concentration at a particular site. It provides bacteria and adhesive effect to tooth surface. So students, in this scenario, all these answers are true when it comes to dental plaque. Don't get confused when you see the time. You might think, oh, it's not 12 to 24 hours, maybe it's lesser or maybe it's longer. Do not get confused and say that this is wrong and the rest of them are true. You have to stick everything as correct. But there could be scenarios where one or two of them will be wrong and the rest four will be correct. So you must know all the information to rule out which are your distractors and which are the correct answers. So if you liked my presentation today, and if you like the way I explain things, and if it is easily understandable to you, please like and subscribe my channel and share it with your friends. And more importantly, please comment below what are the subjects or the topics which you want me to cover so that it can help you. Also tell me if you have any doubts and I will definitely clear them. These are my references. I've already told you that Newbron is a book which is on cariology. I have put some information from that book to help you with your knowledge. So that's all for today. Thank you and keep smiling.